All right, are you ready to get going to introduce our next speaker? Okay.
Ready whenever.
Sounds great. It's my great pleasure to introduce to you all our next speaker, which is Chris Miles. He's coming to us from the Courant Institute at NYU, and he's going to teach us today about mitosis in cells, and he's going to talk about some analysis and modeling. All right, take it away, Chris.
Great. Yeah, thank you all for tuning in, and thanks to Heather and Mason for organizing this session. It's already been a great mix of methods and applications, and I'm glad there's that balance because I'm at one extreme endpoint of that spectrum, which is that I'm interested in a particular data set, and I want to tell you about that today. So where did that data set come from? I want to just give due credit to my collaborators, and all of the data that you'll see today comes from the New York Department of Health. This lab, Alexei Kodjakov's lab, he's been working on mitosis for an enormous amount of time and has done great work and really is a pioneer of imaging, and that's what allowed this project to be possible. And his postdoc Fiore has really been in the trenches doing all of this imaging and really did a lot of the labor of the data set part. And also should thank my mentor, Alex McGillner, for knowing everything there is to know about this topic. I don't know how. So mitosis, this is what I want to talk about. In case you're not familiar with it, it's basically the process of organizing DNA prior to cell division. That's what this video is depicting, so I'll wait for the video to restart one more time. And you can see that there's basically these DNA molecules floating about, and then these green tubes find them and then move them around. And then eventually the red copies of DNA get split apart. And this is mitosis. This is an actual cell turning into two cells with two copies of the DNA. And this spatiotemporal dance of the DNA is the process of mitosis. So it's a really complex multi-part process. And this is the thing that we want to try and understand. So I think it's maybe hard to tell exactly what's going on in that video, so I want to give you a cartoon of the primary components of this process. And I think maybe what you could tell is that there's these two poles, and from the two poles are where these microtubules, these rods or these fingers emanate from. And the first part of the process is basically searching for the DNA that's floating about. So it's sort of scattered about in the cell after it's copied. And these fingers, they grow and shrink stochastically, and they search for the attachment sites, which are kinetochores, and I'm depicting them in yellow. And the ultimate hope is that both attachment sites get attached for each of the 46 or whatever copies of DNA there are. And then after that, after they're both attached, there's these other ingredients that I'll tell you a little bit about, molecular motors that generate forces. And the reason that this is important is because you want to rip apart this copied DNA and split it into two. And I'm depicting them as cars and trucks here because there's a whole zoo of different molecular motors that go into this. And that's why I'm not just depicting them as one type of thing, but many. And really all you need to know is that there's these little proteins floating about that generate forces. And that's why I depicted them as a car. You could think about they want to drive on the fingers and they would pull things apart in the process. So this is a super complex system, and I just wanted to briefly mention that it also has a rich history of theory behind it. Dating back to the 80s and 90s, this idea of a stochastic search, these fingers growing and shrinking, looking for an object, is sort of a tantalizing thing to model. It seems really interesting. Is this a good way to search for an object? And it turns out that if you write down a model of this, it certainly is possible to find all the objects, but if you look at the distribution of how long it would take to really find everything, the model suggests that it would be exponentially distributed, which means there's these things that are sort of living in the tail that take a while to be found. And that kind of makes sense if you think about a stochastic searcher. Maybe there's some things that are going to take a while to find. But in reality, mitosis is this super precisely timed, super robust, remarkably accurate process. And it's definitely not exponential to find and capture everything. It's really more of a Gaussian, and it's a pretty tight Gaussian by cell biology standards. That this process, this complicated dance of things is somehow really, really precise by cell biology standards. And this precision and speed has really been the investigation for the last 20 to 30 years. To give you a sort of unfair summary of 20 years of research, people have basically tried to say, well, what could allow for this process to be both fast and accurate? And there's been a lot of really interesting ideas here that basically maybe the geometry of the things that are being searched for allows them to be found a little faster, or maybe the searching, the growing and shrinking of these fingers is somehow biased by some chemicals in the system. But basically, I would say that all of these things do indeed speed up the process, but none of them really fully explain how this process happens so quickly and precisely. And I don't want to suggest that we're proposing a paradigm shift, but I think the field is maybe a little stuck in some sense, that maybe we're thinking about this entirely wrong, that maybe this process of stochastically searching and then stretching is there, but maybe it's not the right way to think about it. And how do you rethink a system that's been thought about for a long time? Well, you really need a different lens on it. And that's what this project was motivated by, is new data. And that really is what I want to sort of tell you about the most, that I think is the most exciting. Through some biology witchcraft that I don't understand, the lab that I work with, Alexei Kojakov, is now able to track all of the DNA molecules in 3D throughout the entire duration of mitosis. So what I'm showing here, so the yellow dots are the attachment sites. That's really what's being tracked. And I'm drawing a pink connector, sort of showing you that those are the sisters. They're attached to the same thing. And then the gray dots are the poles, which are also moving about in 3D. And I'm drawing a line between them to show you the axis connecting them, but that's not really there. So this is really the first time that we've had access to 3D trajectories of mitosis. And we see that this dance is obviously coordinated, and there's something going on here. And our maybe vague but honest goal was just to say, now that we have this data, can we understand anything different about mitosis? That this is the first time we've had access to this data of this type. What can we learn from it? And that's really the hope that we're going for here. So I want to tell you a little bit more about these trajectories when we look at them one by one, but I need to define some coordinates that really I think make some sense in this context, which is the spindle, the poles, really sort of define a coordinate frame. And what I want to do is I want to use sort of a cylindrical coordinate frame to describe things. So if you imagine the two poles, you can think of that as defining the cylinder. And then I'll define R as the sort of radial coordinate away from the cylinder, and D as the distance away from the center. And the reason that that's important is because ultimately the DNA wants to be in the center. It wants to be in the center so that when it gets split apart into the two new cells, that one of each copy goes to the two new cells. And I also want to define these other two quantities, delta, how stretched the two attachment sites are. And the reason that we want to keep track of that is because if there's really these forces pulling on it to rip it apart, then that should get stretched. And it's known that this gets stretched. And the last thing is this angle phi, which is basically how parallel is the DNA to this axis that I'm defining from the data. And again, the ultimate goal is that it becomes parallel, because if you rip apart this DNA, then you want to go with each of the poles. So if you're not exactly parallel when you rip apart, then this leads to errors, bad things happen. So I keep saying this is what we want to happen, and I'm going to show you an example trajectory of this occurring. Okay, so here is what I would call a typical trajectory. On the left, I'm showing you this dr axis that we've defined from the data. So this isn't the way we get the data. We sort of rotate it so that we can make sense of it and put it in this frame that we think the cell cares about. And you can see what happens here. It goes from light to dark. The trajectory basically just moves and it hits this object that I'm drawing in this yellow oval, which I'll mention later. And basically it stays there. It moves towards the spindle, stays there. And that seems to be all that goes on. But if we look at this D versus time, you see that it does this actually sort of interesting thing. It has these really defined oscillations. So actually what it does is it moves to the middle of this spindle, this pole-pole axis and then oscillates in the middle. So what's going on with those other two quantities I described? Well, right about the time that it reaches the middle, actually this quantity delta, which describes how stretched the attachment sites are, it becomes stretch. So that's good. That means it's being pulled from both sides. And also phi goes to 0. And this is in radians, so it becomes parallel with it. So this is a canonical good trajectory. It ends up in the middle of the two poles. It oscillates, it gets stretched, it's parallel to the axis. This is canonical good. This is everything the cell could hope for. So why am I saying this sort of ominously? Well, there are bad trajectories. And so let's look at this thing on the far left at first. It moves towards the spindle, the poles, but then you see that it looks like it gets stuck with one of the poles. And in fact, this one, it kind of moves with one of the poles the entire time. And well, why do we think this is bad? Well, if you look at the quantity delta, it never gets stretched. It never ends up parallel. And I can tell you that it's obviously not included in this movie, but actually the cell dies when trajectories like this happen. So this clearly is not a good thing for the cell. It basically says that sometimes it does make mistakes. And there are these error correcting mechanisms, but what we hope to say is what's causing these mistakes, or can we understand anything about why these mistakes are occurring, or anything like that. So even before that, I mean, there's this really sort of basic question of, I'm sort of telling you that this is a bad trajectory, but is this really the most sort of natural way of splitting the data? So I think for people that know data science, this isn't really that novel, but I at least want to mention that we did it. You can go through each trajectory and assign it a barcode based on these sort of human-defined characteristics. Does it oscillate? Does it end in the middle? And these are from basically biology folklore, that people have observed these in years past, and they think this is what defines good and bad. And if you assign each trajectory a barcode based on these features that people say defines good and bad, does it really? And you can cluster all of the data into 2 clusters and make sure 2 is sort of the right number. And you see, lo and behold, yeah, that sort of the most natural split of the data are these ones that end far from the center and don't oscillate, or end in the center and do oscillate. And you could say, well, what happens with the other features? But I don't want to dwell too much on that. But I mean, in some sense, this is just a sanity check, that there really are good and bad trajectories as far as the cell is concerned. But I think what's maybe most interesting is if you look at where these come from. And I said I pooled all the data together. And so what I want to look at is this histogram for a moment. And in healthy cells, so green is good trajectories and purple are bad trajectories. And what you see is in healthy cells, there basically are no mistakes. And that's good, right? You would hope that healthy cells can do mitosis basically error-free, and that's definitely what we observe. But the biologists are able to basically disable some of those molecular motors that we know are essential for this process. And what they can do is disable different types of molecular motors. And you can see if you disable this first type of motor, which I'm labeling CAR, a bunch of mistakes occur. So that says it's probably a critical part of the process. If you disable the one that I guess is a trash truck, you get some errors, but fewer errors. But this totally bizarre thing that we still don't understand, but I just wanted to share with you because I find it really exciting. If you disable both of them simultaneously, the error rate is actually less than if you just disabled the CAR alone. So this says that somehow these things are working together, maybe. And this result, it was sort of written down in the folklore of biology a long time ago, but I think this is the first time that we've actually at least sort of been able to quantify it. And we still don't have a good explanation for this, but it at least convinced us that something interesting is going on with these trajectories, that we can learn something interesting. But I mean, note that at no point in this analysis do we really understand any mechanisms. I mean, we decided what defined the characteristics. So we wanted to say, now that we have these trajectories, can we just really learn something from the trajectories themselves? So go back to really what the data looks like. And okay, if you think about particles moving in space, especially in a biophysical context, what people often do is they characterize the motion. They say, well, was this particle diffusive? Was it subdiffusive? Did it switch between these? And I think this is a perfectly reasonable, even good approach for a lot of things in biophysics. But in ours, it doesn't really make sense. I mean, we really care about space itself, that there is this coordinate frame that really is meaningful, and we want to know, where does stuff go? Where does it move? We don't, I mean, of course we care how it moves, but we really want to know, where is it moving to? And that really isn't captured in this sort of classical analysis of biophysically moving things. So I found this idea in, I think, the voice recognition literature. So this idea of how do you basically compare time series? And if you take two time series, you could think of them as vectors and just compute a Euclidean norm between them. And this is fine for a lot of things, but it suffers from this issue that if the timing is a little bit different between them, I think Siri is angry at me for some reason. We'll let that go. If you look at if the timing between the two time series is different, this leads to huge errors in the Euclidean norm. So there's this really cool idea. I don't know how to disable that. I'm gonna just close my screen. Okay, it has gone away. All right, sorry for that technical blip there. Okay, what I was saying is dynamic time warping. It's this way of comparing two time series. And what it does is it basically allows for the timing between two time series to be different. And the way it does this is it tries to do a minimization between the distances between two time series, but it allows for time shifting between them. So what I'm showing in this bottom panel is that the red and blue time series are basically the same, even if the timing is a little bit different. And the minimization problem you do to basically compute this distance is you allow for the time points to shift a little bit in time. And this is a really natural choice, I think, for cell biology, because although I said the timing is precise for cell biology standards, cell biology is not a very precise thing. And timing between cells, between chromosomes, really varies quite a bit. And we don't want it to. We want it to really not penalize that. So we want to use this dynamic time warping idea. And what we can do is we can now look at all these trajectories in the RD coordinate system, this cylindrical coordinate system. So remember, D is along the spindle poles. So D equals 1 would sort of be at the pole, and R is away from the poles radially. And what you can do is you could say, okay, well, with this dynamic time warping as a metric, basically comparing trajectories, what are sort of the typical trajectories? And to be honest, I don't know that these are all that insightful, or looking at them, you can really learn that much. So we have this one in pink that looks like it moves and then sort of stays, like the first one that I showed you. There's this one in green that starts kind of near a pole, and then it moves along some surface and ends up in the middle. And then there's this kooky blue one that sort of moves away and then close to it. And that one certainly seems to be the most exotic. So if these are the centers, after doing some clustering on the raw trajectories, do these have any meaning at all? And as a sanity check for that, we just said, okay, well, if you look at how much these oscillate, is there any difference in the ultimate fate of these? And I guess what I'm saying is we don't have a decisive answer. Siri is still quite angry at me. We don't have a super decisive answer, but we can at least say that those blue trajectories seem to oscillate far less than the others. And maybe that's an indicator that the trajectories themselves can tell us about the ultimate fate of the chromosomes. That was our hope, right? That just being able to track these things in 3D, we can somehow predict their ultimate fate. Is it a good trajectory or bad just by looking at how it's moving? And this really is saying that, yeah, maybe this is possible. That the things in this blue cluster really do seem to oscillate less, which is a proxy we know for being a bad trajectory. So this proof of concept that the trajectories themselves really do encode their ultimate fate in some way. You'll note that in this, they seem to hit some sort of object, a shell around the poles. And what is that? Well, if you look at where they end up stretching in this coordinate frame, it's sort of clear that there's some sort of oval, some sort of ellipsoid that they seem to hit and stop. And we went to the biologist and we said, what is this? Like, why do they seem to stop? And they were like, oh yeah, of course, there is like an actual ellipsoid around the spindle defined by these microtubules. And they showed us this picture and they're like, yeah, it's exactly that ellipsoid that you're seeing. But we really didn't understand why. I mean, why do they hit this thing and then seemingly stretch and align? And this is where we wanted to use more classical modeling to try and understand what we're seeing. That, okay, basically what we've learned is that the good trajectories move towards the spindle, hit this ellipsoid, and then become stretched and become parallel with the axis. Why? Why is this? So we thought about it. If the DNA is moving towards the spindle, if it's not at the ellipse, what's sort of different? Well, the only thing that's really different is the angle at which the microtubules are basically aligning. That if you think about them coming out radially from the poles, then this angle that I'm calling gamma really is maybe the only thing that's changing as you get closer and closer to that ellipse. So can we come up with a maybe classical biophysical model which says why changing that angle gamma might lead to alignment and stretching of the DNA? So I don't want to talk too much about the model, but I basically tell you these ingredients that we modeled the two attachment sites in a classical biophysical model. So there's springs attaching them, which we're going to look at the thing stretching. And then it's just a sum of forces on those objects. And the forces are just determined by these attachments that they would make with the nearby microtubules, and everything is a spring, basically. The molecular motors, I said they're force generators, so what do? They try and walk, they try and generate forces, and it depends on the force that's on them. It's not that exciting, but the one sort of interesting feature that we wanted to include is that the attachments can break. That if you think about tugging on these attachments, that would be what's allowing it to attach to the microtubules. They should break as forces are attached to them. But here's sort of the ingredient that we wanted to include that we think is an interesting idea that's sort of obvious when you look at it. If you have an object and you have attachments coming out of it, then ones that are sort of coming out of the object should be good attachments, that it's sort of reasonable to think of things poking out of the surface of that attachment site. But this top object, which would sort of be like an attachment bending backward, should be pretty bad. So the way we have it as bad is we just have ones that are sort of not in the same direction of the surface, breaking very quickly. And otherwise, we just let this thing stochastically evolve, and what do you see if this happens? Okay, so in this bottom one, I want to show, imagining that you're on the ellipse, and my computer seems to have frozen, which is, okay. So on the ellipse, what happens? Well, you could think about the microtubules being parallel, and you can see that, okay, these attachments form, but ultimately the cargo ends up pretty stretched and parallel with the ellipse. And finally, if you think about, well, if you were away from the microtubules, or away from the ellipse, what would happen? Am I having some technical issues as well with this? All right, can I play this video? All right, well, for the sake of time, that video does not seem to want to play, but ultimately what you find is that it doesn't align with the ellipse and it doesn't become stretched. So we sort of said that this classical model in some way describes how hitting the ellipsoid would really work and why it's a meaningful thing. But okay, so what have we learned from this? What we learned is that if you look at trajectories one at a time, you can learn stuff from them. But the exciting thing about this is that we have all of the trajectories at once that are occurring in the same cell. And we haven't really done due justice to thinking about these things coexisting. And that's really what we want to do next, is do they interact in any way? Of course, we've said something if they evolve one at a time separately, but we want to know something about them evolving together. And given that this session is about TDA, and this community especially puts out incredibly documented tools that even someone like me, which knows relatively little about this, can do, I thought, well, okay, this seems like the right thing. If you think about these particles evolving in time and space, let me compute these Betty numbers. And just to do sort of a quick, I mean, there's not much going on this slide except to say the rows sort of indicate different cells of the same type. And I was hoping that when I did this, the rows would look immediately different. But if you look at this and squint at it, it's not really clear that the healthy row is visually different than the say one motor knocked out row, right? I mean, if you squint, you see, well, maybe there's some differences, but I was really hoping that there would be an obvious difference. And I know there's a bunch of TDA experts listening to this that have done this successfully, and I would love to hear, maybe I'm doing something dumb with this. I'm happy to talk more about what I did. But in the future, we really want to go this direction of trying to think about it as this many-body interacting particle model and use machine learning to either back out what the forces between the particles are directly, or use TDA and machine learning in some junction, like people have done successfully, to say something about the collective motion of these chromosomes rather than looking at them individually. So just to conclude very quickly, I hope I've convinced you that this is an interesting application that's ripe for the development and application of tools that either already exist and I don't know about them, or need to be developed. And I think it also is a classic case that shows that classical modeling and sort of data science can exist in harmony. And yeah, yeah. So thank you. I appreciate everyone listening and tuning in, and thanks.
Hey, thank you, Chris. So it looks like we already have a question from Veronica Chocanel. So Veronica, go ahead and unmute yourself.
Oh shoot, I didn't realize we were doing this live. Okay, hi Chris, this is Veronica. So I had a couple questions. First, I was wondering, what is the motor that you find to be critical in your analysis? And second, in the case where the experimentalists disable both motors, is it possible that there's something else other than the trajectories that is bad about that instance of mitosis?
Yeah, both incredibly good questions. If I can go back without my computer getting angry. Sorry again, all the technical issues. I have no idea why they're happening over Zoom today. Yeah, so the motors, if you're familiar with this topic, it's kinesin and dynein are the two types of motors. So sort of these classical two families of motors. And yeah, so your second question, when you knock both of them out, you could also look at sort of how many of the cells end up dying. So if mitosis doesn't happen properly, the cell just dies. It just gives up and says, I can't divide, I'm gonna die. And also, if you look at sort of how many cells die for each of these four categories, it also sort of reflects what I said. So yeah, something else could be happening, but at least this breakdown of knocking out kinesin alone is worse than knocking out both. It seems to be true even at the sort of cellular level of them dying. So yeah, we hope to understand why this is, but we don't have much yet. Yeah.
Got you, great, thank you.
Yeah, thanks.
And again, if we have time for more questions, if you have a question, just ping either me or Mason in the chat, and we'll call on you to ask your question. Are you seeing any questions on your end, Mason?
No, I don't see any right now.
Veronica said she has one more. So
all right,
we're waiting to see if we
let's
have
do it. Yeah.
I'll keep going. I completely missed what you were applying TDA to, like what part of your data.
Yeah, so I went through that quickly because I sort of thought of it as a failed experiment, but maybe I can go back to that. Yeah, so I mean, we have these particles floating around in 3D as a function of time, and I was just thinking about them as actual particles to do TDA on. So draw the ball around it at a fixed time, and then looking at what the Betty number is at a fixed time. And then the x-axis here is how it evolves with time. So I was hoping that if you look at all of the 46 particles, the DNA particles at a given time, and do TDA, get the Betty numbers, the evolution of that in different cell types would look different. But yeah, it's on the actual DNA particles, the 46 of them.
Yeah, so you were doing the Crocker plots through time.
Yep, yeah, exactly.
Yeah. Okay. Thanks so much.
Yeah, thank you.
Okay, so we have another one. This is from Jose Carrillo, and he asked that I ask by proxy for him. So his question is, how are you planning to model the forces or parameterize them?
Yeah, this is a thing that I would love suggestions on. I mean, naively, what we've started doing is just repulsive or attractive. Just do things want to stick together, or do they repulse each other? Because we don't even have intuition for that, for the DNA particles. We think they might repulse each other because they're so big, but that's sort of where we're starting with. But I would love to hear any suggestions of ways to parameterize the forces to make this more tractable. Yeah.
All right, I think we'll probably stop the questions there. So let's thank Chris again and get our next speaker set up.
Thank you.